So as he said, my name is Joshua Johnson, and today we'll be going over my project that has to do with influence of flower openness on yield and cotton. So just the introduction that everyone has to give. So cotton is considered the world's most important textile fiber crop. Out of the 50 recognized species, 45 are diploid and 5 are tetraploid. Gossypium hirsutum, which is upland, and Gossypium barbadense, Pima, are both domesticated tetraploids. So to go over some U.S. cotton production, Texas is the largest producer, which approximately 40%. Most production is concentrated in the high plains, as you can see in the figure to your right. Estimated value is close to $7 billion. We're talking about Lent plus yield in 2019, and U.S. leads the world leading exporter in cotton, comprising about 35% of the global total. So jumping into some project objectives. So I'm collecting phenotypic flower data on about 15 experimental lines, determining if flower openness is going to influence lint yield, and develop some experimental lines with different flower openness to be used in later studies. So since my project is mostly on flower openness, I thought I should go in with some cotton, talk about the cotton flower. So the cotton bloom is a perfect flower, which means it contains both the female and male parts. Floral buds appear as small green pyramid-like structures, and in the cotton world, we like to call them squares. Time for first square until anthesis, which is flowering, is about 25 days. And at first, the petals appear to be creamy white. And then the day after, they turn into a pinkish red, and then they get to a more reddish color the further you go. So this is just a picture to show you. So on day 1, flower is pretty white. Day 2 turns into a pinkish color. Day 3 to 4, pinkish red. And then 5 to 7 is when the flower starts to shrink. So diving into what some potential importance of flower openness would be. So a lot of major crops have sensitive reproductive organs, cotton being one of them, and because the pollen is considered pretty fragile, and that's why we're also looking into the effects of pollen sterility. Pollen is susceptible to pollen dehydration and osmotic lysine, which, if you've ever seen it under a microscope, it's pretty cool because once the pollen sacs interact with water, within a few seconds, they burst. In many environments, an open or closed flower shape could affect the flower's microbiome. So this is to help people understand really what we're looking at here. After putting these in, I realized I could have used a little bit of better pictures, but I think these will get it done. So on the left, you can see one that we depict as being open. Another way that we like to talk about flower openness is how much the petals overlap. So you can see on the left in the open one, there's little overlapping, and it seems to be like a wider angle is what we're looking at. When we talk about the middle picture, an intermediate one, petals are overlapping a little bit more, and we're getting a more upright position. And then in the right, we're talking about closed. You can see the petals are overlapping a lot, and we see that it's in an upright position. So some traits of interest. Common cotton measurements that we take are micronair, fiber uniformity, fiber length, fiber strength, elongation, and yield. But the ones that I'm specifically looking at would be the flower openness rating, pollen sterility, seed per bowl, and the variability of flower openness within the line, which we will talk about in a little bit. So calculating flower openness rating, since this is a pretty new, novel idea, we definitely went through a lot of trial and error, trying to figure out what would be the best way to measure this. I think we went through 4 to 5 different ways. And finally, we just went with the KISS situation where it's keep it simple, stupid. So we, because we just wanted to get a reliable way to get empirical data, because the only studies that had ever looked at this in the past were mainly just taking pictures of flowers, giving it a 1 through 5 visual rating, and that's as far as they went. So we wanted to get empirical data that we could analyze through conventional approaches. So that means that we would want a measurement that would actually give us a consistent angle, and we're measuring this in degrees. So we decided to go with a method that gave us 2 different measurements for each flower, and then employing some trigonometry. So the first measurement that we would be taking would be petal length, which would be the hypotenuse. And the second measurement would be the middle of the flower to the middle of the petal edge, which would be the radius. And I have some visualizations to help understand exactly what we're talking about, because it can be kind of confusing. So the measuring specifics, we went into Excel and using some trigonometry functions. Since we want degrees, we use degrees, a sine, radius divided by petal length. And this would end up giving back to us the angle of the flower that we're measuring. So down here with this illustration, we can just see the opposite if we're talking about Stochatoa here. Opposite would be the middle of the flower to the middle of the petal, which would also be the radius. And then we have the hypotenuse, which would be the petal length. And then plugging these 2 numbers in, it would give us the angle. So these are some of the methods we ended up trying. We have the digital calipers, which ended up probably being it was the most efficient way by far. We tried out this digital angle gauge. It's just not you can't calibrate it very well in the field, especially when stuff is moving around pretty easy and it gives inconsistent numbers. And we just had regular calipers, but the digital calipers definitely turned out to be the most efficient when you're trying to do this fast. So here's just a better picture to help you understand. So B would be the hypotenuse that we're looking at here for our measurement, and then C would be the middle of the flower to the middle of the petal. So another underlying problem that we ran into is because breeders haven't really been looking into this. Flower openness is not a trait breeders have been really paying attention to or selecting for. So when you're walking through, especially my experimental lines, some of them, as you'll see in the data a little bit later, are highly variable. And it's the same line, but you can have a really open flower, and you can also have a really closed flower. So we had to go in and develop a flower variability score, which we measured 1 through 5. 1 being pretty stable and 5 being highly variable. So when we're talking about data collection for the flower openness, we wanted to make this as consistent as possible. So we tried to we did collect data in between 12 and 3 p.m. We started out doing trying to do a shorter amount of time, but through trial and error, we just needed more time to get the amount of data. Data collection, of course, if we started the rep, we had to finish in that day because depending on the weather and cloud cover, the variation of flowers can change. And then we, of course, took daily high temperatures, relative humidity, and cloud cover, because they've seen that it can affect flower variability. We, of course, like I mentioned earlier, we had to walk through each line before we went and took ratings to get the flower variability scores for each line. And we got 10 flowers. We tried to get at least 10 flowers per plot to get the measurements. And then they were tagged with ribbon for each flower that we measured. And then when the bowls matured, we went back and hand harvested them. So the other part of my project is trying to measure pollen sterility rating. So we also did a 1 through 5 scale on this. And this is just trying to see how fertile the pollen is, because it does vary in between the plants depending on the weather or any other environmental factor. So 1 is 100% fertile, 2, 25%, pollen sac non-dehiscence, 3 would be 50%, 4, 75%, and then 5 is totally sterile. So we are also implementing a greenhouse study. Unfortunately, the data is not done being collected for pollen sterility, flower measurements, so I will not be presenting that today. But it gives us a good way to give pollen variability, because in the field, it's super hard to measure just because there's so many different environmental factors. And we also use the greenhouse study to advance some of the lines. So environmental effects on pollen, high humidity is a big one, only because it causes the pollen to become sticky, is a way of saying it. and it prevents proper dehiscence, so the pollen's not able to move around or be moved by insects as it normally would be. Rain, as I mentioned earlier, has a problem. If it comes in contact with the pollen sacs, it will lyse or burst. And high temperatures can also dehydrate the pollen, and that makes the pollen fatally destructive. So one farming tip that I have is using sprinkler irrigation systems will likely destroy your flower viability the day of flowering. So if your crop is flowering, I would not water it only because it affects the pollen so much. Jumping into our experimental design, field experiment, dry land irrigated trial, 15 experimental lines, 1 conventional check, 10 caught 73. Randomized complete block design with 4 applications. In the greenhouse experiment, we had the same populations that we used in the field, and we had a complete randomized design with 2 applications. So for data collection, 5 days, we were able to get out there and do it. I definitely got faster. At the very beginning, I was moving way slower, but by the end, knowing what I'm doing, it went by way faster. So 5 days from August 9th to August 13th. As we can see, humidity is pretty similar. The daily temperatures at the time of recording data were pretty similar. Cloud cover, we had some discrepancies, but day 5, we did have a cloudy day. So that was a major difference. We only collected flowers on repetitions 1 and 3 in the dry land and irrigated trial. And so in total, we have about 640 flowers that are being measured. Jumping into some results, we have the dry land yield trial. So as we can see here, we do not have any significance in treatment. We do in rep, it's highly significant. We have an R squared of 0.712, a CV of 16.77. And then this yield, the distribution of yield is the average yield per plot in pounds. And these are all the different cultivars that we are using. I'm assuming there is some wide variability, but we're also just working with 1 year of data. So that's what I will contribute that to. Again, for the irrigated trial, you see the same thing where rep is significant. We have a lower R squared of 0.37, a higher CD of 19. And again, when we look at the distribution of yield, it is variable, also due to 1 year of data. Now, when looking at flower openness, when comparing flower openness to the genotypes being used, we do see a highly significant for treatment. So we do have a lower R squared, CV of 26, which is pretty high, but we got a flower openness mean of 29.64. Like I mentioned earlier, so we see right here that wheat WK11 low in red. Whenever we look at the flower openness, it is highly variable, which is just something that we saw on multiple lines. But then when we look at 17 SJ47, we can see that it hardly has any variation, and we also have other ones that have low variation. So one thing I wanted to do for my project was recognize the groups. So the first thing I did when I got the data was just run it in Excel, average all the flower openness from each plot for the genotypes, and I got a ranking. As you can see there, 1 being the most closed at 22 degrees, and 16 being the most open at 36 degrees. So those are the rankings. But then, for the flower openness, we used LSD, and we got significant groups. And as we can see here, we got 17 SHJ48 and 17 SHJ55 standing out a little bit. And then at the bottom, we have 17 SHJ32 standing out a little bit. So for so they tended to overlap a lot, especially in the middle of the data. But I wanted to separate them out into 1 through 5 groups so that I could, because I needed to define the potential parents to make combinations that will generate homozygous extremes for additional experiments. because I just want to see the variation when we make crosses, what we will get. So this part, there's going to be a lot of numbers. The next one I think is a little bit better, but we can see here that we have a lot whenever we're just looking at correlations here. Whenever we see the flower openness compared to all the other normal variables that we're looking at, we get a lot of negative correlation. We have these two positive correlations and much more negative ones. And this is going to be the same thing, just better visually. Where this is a correlation matrix heat map. So what this is saying is if it's a blue square, it's negatively correlated. So the more blue it is, the more negatively correlated it is. And if it's the red squares are positively correlated. So we can see that we mostly have negative correlations. And then we get to seed cod weight and lint weight, where we have very small positive correlations. But then when I ran some significance to get some p-values, the only one that even stood out that was significant at all would be elongation with floral openness. So then I want to run a PCA just to dive a little bit more into the data, just to look at it. And we can see here that we have some groups of data. So we have what would normally be considered quality traits on the top right, and we up in for yield traits we got in top left. And we do see generally, if you're breeding for quality, your yield is going to go down. If you're breeding for yield, your quality is going to go down. Generally, it's not always true. And then we have what I'm actually looking for here, what would be flower openness. And it is all the way down here by itself in the bottom, which would tell me that when we're selecting for flower openness, we're most likely going to be negatively impacting yield and quality traits, which is a story to tell, not exactly one that I wanted to see. So we want flowers that are not just completely white, and then that's what I get from this PCA. But another thing to mention is my principal component 1 and principal component 2 is only showing about a little over 40% of the variation being explained. So in conclusion, in both the dryland and irrigated trials, we saw no significance of monogenotypes. Flower openness was significant, but as we saw, flower openness definitely, from the little amount of data that we have for this 1 year, inversely related to many of the beneficial agronomic traits that we would be selecting for. So for future research, definitely want to gather more data, at least double the amount that I had at this point. So for this year, and test significance and correlation to see if they still match up. Make crosses within the grouping, the 4 openness groups that I did make. I want to cross open and open, close and close, and then open and close, just see the amount of variation we can get, and then determine the heritability of flower openness. So these are my references. Thank you, and I would like to thank my committee, of course, Dr. Hague, Dr. Deaver, and Dr. Sport. Of course, the Texas A&M Cotton Improvement Lab, Marshall, Paige, Jake, Alexis, and all the undergraduate workers that help us get our work done. Texas A&M Stone Crop Science Department, Cotton Inc. for funding for the project, and Texas A&M Agricultural Research. And with that, I'll take questions.
So did you image these flowers? I know you used the calipers, but did you image them?
We did not image them. That's what they did in the previous ones. That would be an interesting idea to do, to make sure our measurements would be, to see how they would compare. Only because, I mean, they're being out there, we're trying to move pretty fast. So the calipers was probably the most efficient. I mean, it was the most efficient way. And so imaging would be a good idea. It's something that I've read in previous papers.
I think you can increase your throughput and also your objectivity by doing that. But there's also a lot of image data sets out there. And if you threw that out there, you might find some computer scientists that could help you get some more objective measurements.
Yeah, it definitely takes a long time. And it was a bunch of trial and error just to get the data to begin with. Thank you.
How is the openness affecting fiber quality, like length? How do you make that connection? What's happening?
Oops. I mean, in the correlation that we ran, it just seemed to negatively affect most of the quality traits.
But is it real?
Was it significant, right? So
What'd you say?
Your correlations were not significant, so they weren't.
So there's no way. Yeah, the only one that was significant was elongation. So.
Did you measure the same flower position on each plant, or how did you?
Yes, sir. By the time that, due to unfortunate circumstances, by the time I got out there, it was mainly from the middle to the top of the plant. So we tried to stay in the same position of the plant to make it more consistent.
I really appreciated that you showed the temperature on the days you were out there that was 90. How did you find cool days of 90 to be working in the cotton field? When I was out there, it's always 100. But,
I mean, it definitely feels like that.
I would say once upon a time, we did a lot of selection for open flowers.
Mm-hmm.
It happened to have been with Pima because we were looking at hybrids and we wanted them to be open for bee activity.
Yes.
So we not only stared at the bee activity, but we selected for openness. Not sure we noted the negative quality impact.
Yeah, that's interesting too.
That was in New Mexico as opposed to humidity belt of College Station.
So I have a question that's similar to Dr. Parker's progress. From a gene flow standpoint, the flower openness might affect one way or other. He touched on the insect mediator, bee mediator gene flow that maybe potentially flow in movements, the openness going to have an impact.
When I was out there was definitely a lot of insects. Actually, one in particular was fire ants. I guess they love being in cotton flowers. So I got eaten up by them a good amount. But I think having a closed or open flower, if the insects want to be there, I would think. But I still think an open flower is going to be a better way for insects to actually get in there and move around pollen. But other than that,
can cotton be being pollinated?
No.
Did you see the tornado yesterday? Maybe. But.
Yeah, it's not known to be much pollen.
Just FYI, the line that you showed that had so much variability within it, the openness, that line was actually developed in mass selection. So when I was putting that line together, I was just grabbing plants that had the same length, shape, and color. And so there should have been a lot of variation. The other ones were derived from single plant selection.
So how many genotypes did you use in this? And how were they selected?
16. And I guess in a lot of different ways, but they don't have that similar pedigree. So.
Well, and that's sort of what I'm wondering is if your negative correlation in part is due to sort of cryptic population structure of what was selected. Just if you had like a recombinant inbred line population or some random mating population that would break up those linkages, maybe that's why he didn't see the negative correlation. But
makes sense. Thank you.
Other questions? Senator,